This is Gigaohm News. Welcome to Gigaohm News. This is high resistance, low noise news brought to you by a biologist. My name is Jay Cooey. I live in Pittsburgh, Pennsylvania, and I'm going to try and bring you this news stream once a week on Saturdays and between 2 and 3 times a week during the week when I have time. But Saturday's stream will be the important one because Saturday's stream will be supported by a PDF document which will give you some links to the papers that I discuss, along with some annotation, some discussion that will make it not necessary for you to take notes while we're having this discussion on Saturdays, because the PDF will provide you with that already. The PDF that I am now referring to for today's lecture is not quite ready yet, and that's because I have been working on a video for you. As you know, there are other things that my channel does. One of the things that my YouTube channel does is it does videos about neuroscience and other things like that. This video is actually a bibliography video which covers a lot of the papers that I'd like to talk to you about today. And so it's just kind of it's a production video complement of what we're talking about today, with a much less, with a much less formal attitude, and certainly no chance to ask me questions while I'm on the bike. So anyway, I just wanted to let you know that that's going to be released tomorrow as well, so check that out on YouTube. But let me just get started by saying that I think one of the most important things that we need to talk about right now is establishing sort of a common ground between you and me. If I'm going to play the role as the information provider, I hesitate to use the word or the term teacher, but it's how I feel in my heart. What my gift is being a teacher, and so I'm going to use that word, even if you don't see me as a teacher, but you see me as a peer, someone whose opinion and insight matters to you, even if you don't take all of it, but you take everything with a grain of salt. It's nice to have someone whose opinion matters to you, even if that opinion is not always right, as long as you know that I'm the kind of person, the kind of entity that's going to correct himself when I get it wrong. That's the kind of biologist that you're interacting with now. So as your instructor on Saturday afternoons, should you let me be that instructor, what I'd like you to do now is just try to come with me into a particular place so that we understand the common ground on which we stand, so that we move forward together with a common purpose. The narrative that we are being told, and sort of the thing that's going on right now, started already 20 years ago when we were kids. I'm 48 years old, so when I was a kid, we're talking about the late 70s, early 80s, where the adults in my life were already talking about the fact that the media was starting to become biased and controlling and manipulative. And even though there were only 4 or 5 major networks and only 4 or 5 TV channels when I was growing up, when I still had to stand up to touch the television to change channels, and that was okay because you only had to search between 2, 7, and 13. These 3 channels were already known by adults when I was a kid to have been sort of biased, and the information was controlled, and that controlling that information had advantages for people in control of that information and could manipulate positions of power, it could influence decisions, etc. So if adults, when we were kids, were already skeptical of this, I think it's very important for all of us to consciously acknowledge, that the situation that we're in right now is not any better. The situation that we're in right now is not any better than it was when we were growing up. And in fact, it has to be, by definition, just because of the number of television channels and the number of social media outlets and social media tools, traps, algorithms. It has to be, by definition. You don't need figures and facts and numbers for me to tell you this. Just like you don't need me to explain to you that when you were a kid, there was more snow in the winter than there is now. All of us who are over 40 years old know that. We don't need anyone to tell us that, but the television is telling us something different. Social media is telling us something different. The separation of social media from the sort of regular mainstream media is in and of itself a construct. It is a falsehood. You shouldn't do that in your head. You shouldn't think that Facebook and Twitter are any different than CNN and MSNBC. That for sure not. These are all one collective set of psychological tools used to rule a nation of people. Used to rule a nation of people who are social, a nation of people who are dependent on money, a nation of people who are largely connected digitally rather than physically. We know this is the strategy, and we have to start here together in order that we all acknowledge together that there is a certain amount of weeding out that needs to be done. Your consciousness is prime real estate. That's not my idea, that's someone else's idea, but it's a god dang good idea. Your consciousness is prime real estate. And corporations and big piles of money, including banks and private equity firms and multinational corporations that are funded by piles of money, all of them are taking advantage of this system in order to game it for themselves. Misinformation is a misnomer. Fake news is a misnomer. And listen to this very carefully. Your consciousness is prime real estate. This misinformation is not misinformation. That implies some kind of incompetence, some kind of random tomfoolery. No, all the information that you see, all of it on social media, all of it in the mainstream media is carefully crafted. Words are carefully chosen and repeated. Words are omitted carefully and often with intention, folks. You cannot look at social media and mainstream media. You cannot look at any of these sources of information. The fire hose is wide. The water's coming fast. You cannot look at any of these sources of information as objective. Even I'm not objective. I am biased because I have 3 kids and a wife whom I care about more than anyone else in the universe. So you're damn right I'm not objective. But my bias is not based toward billions of dollars of profits for entities behind me or those that are paying my salary. So in some ways I'm less biased than other sources who are giving you that information. And that's the problem right now. Is that what's happening is that there's a twofold deception about what's going on. It's basically twofold, and I'm going to help you with one side of it. It's a twofold deception, which is based on money and how you think it works, and biology and how you think it works. And they deceive you on both of those, and so you are on ground that is unstable. You are playing a game for which the rules are not fair for you and are allow cheating for others. And most of us, like 99% of us, are trapped somehow or another in this web. Even I, who am coming to you with some message of I can help you get where I am still trapped. I am still a victim of misinformation. Some of the things that misinformation is again a misnomer. I am a victim of this of the amount of false information available. And so one of the things that I think I am perhaps more skilled than average at is reading and sorting out that relative to from a biological perspective. That's what I want to try and offer you on Saturdays. That's the middle ground. That's the common ground that we have now. I can't help you with money. I know a lot about it. I know a lot about the deception of fiat currency. I know a lot about the manipulation of stuff, but I don't know enough to tell you where to put your money. I don't know enough to tell you how to invest your money, and I don't know enough to explain to you how all of these things work and are these instruments, financial instruments, are intertwined and how they interplay, mainly because I know that money is added to the system in an unchecked and unfettered and unregulated way, such that if you want to understand the way a resource like money affects the biological function of a system, you need to understand how money is limited, and I don't understand that. I don't understand how the Federal Reserve can print money and what are the consequences of it, other than my vague understanding that if you print more, then the stuff that we have is worth less. But from a biological perspective, the amount of deception that they use from a biological perspective in order to perpetuate this system where the money can be weaponized against you. The biological part of this deception is what I want to help you break. And that the reason why is because I believe if we break the biological part of the deception, then the monetary part is just math. But as long as we are afraid and unable to critically think about our own biology, then we will be unable to critically think about our socioeconomic situation, of course. Like if you're terrified to die, you're definitely not going to figure out how you can afford organic food. And so what I feel my first and foremost mission here in the next few weeks is to make sure that everybody who's watching the Gigaohm news stream and everyone that you share it with is going to stop feeling like they might die. And if you feel like you might die, it won't be because of some phantom that you feel behind you that has been generated by social media and the television, but it will be because of biological facts that you understand, that you have a grip on, and that you can take control of and make changes in your life to minimize. That's what I want. That's what this channel is going to do. So what I want to say today, and the information, the sort of idea that I want to give to you is a positive one. Always a positive one, even though I'm serious and I'm always squinting and I'm, crunching my eyebrows down. I'm a happy guy. I got kids that I love. I got a wife that I'm so lucky to have. I'm a happy guy, but I'm terrified of what they're doing to the biological story of the world, what they're doing to the biological narrative that describes our world, because my kids are growing up in it, and I'm a biologist bringing them up in this world, and somebody is changing the narrative as we speak. That bothers me a lot. And what is that narrative that we're talking about right now? The narrative is about a coronavirus. And the narrative is the idea that the coronavirus was released or escaped or penetrated into the human population sometime in November or December in China, and then escaped around the world and is killing and infecting millions and killing thousands. The narrative includes the idea that even healthy individuals can develop very severe symptoms. The narrative also includes the idea that there are people who can develop long-term, long-haul COVID, where they can be sick or tired for weeks, even though this could be a number of other diseases. If you have the RNA test and you have it, then it's, of course, put in the COVID bucket. The other part of this narrative that's very important to keeping the narrative running is that asymptomatic people can spread the disease. Asymptomatic people can actually be super spreaders, has been said many times. And asymptomatic people should feel like they have to protect others. So since you can't know if you're sick, everyone has to assume they're sick. This is a spectacularly new biological way of thinking. Another part of this is that kids can get it and spread it. 100% true. It has to be, because biologically speaking, that's true for every single pathogen in history. So you don't have to worry about that. It's not that kids can't get it and spread it. It's that kids don't really die from it. And dying is important. Masks and physical distancing can slow the spread. 100% true. But slowing the spread can also just mean that more victims in the future. Slowing the spread can delay the end. Slowing the spread can mean, in theory, a more thorough and complete infection of the population than if we didn't lock down. And that's hard to understand, but it's totally and completely 100% epidemiologically true. And closing schools, this is finally the other thing, right? The masks and the physical distancing will slow the spread. This is 100% true, but will it save lives? This is 100% unproven. We don't know if it's saving lives because these people could just die in 4 months instead of this month. And instead of having 1,000 people die in a hospital in Pittsburgh in a couple months, we may have hundreds die each month for the next 8 months. And in the end, the same 10,000 people will die. This is a mathematical possibility, and in fact, a very high mathematical possibility. I would go so far as to say that in New Zealand, that's what you're seeing right now. They locked down. They never had coronavirus endemic yet. It wasn't really there. Now it's there. Now they're locking down, but that won't matter. It'll just slow the spread. New Zealand may be locked down for months because they're always going to have more positive cases. It's impossible not to because the test isn't accurate. So then we get to the point where are we? Well, we are 7 months in. They closed my kids' school in the end of March, and they have not opened since. We should have opened this week, sorry. We've delayed our opening for 2 more weeks, at which point we will open 100% remote for the first 4 weeks, trying to decide what to do from then on, but I assure you we will remain remote. So what to do now is the real question here. Where can we go? Are we running on a wheel now? Because the death toll is not increasing like Fauci said. The case number went up, and it's going to continue to go up as we test. But the death toll in Houston went up and spiked and went down and is gone. The death toll in Florida went up and is gone. The death toll in Arizona went up and it's gone. It's not that people aren't still dying, but they're not dying in the hundreds anymore. They told you yesterday that we had the largest single-day death ever, but remember that the CDC moves around dates of death and dates of reporting. And so even the reporting is something very difficult because, again, there's too much disingenuous reporting of the numbers. And that disingenuousness is used against us on the news and in social media without qualification. As a scientist, I'm used to hearing numbers with a sort of standard error, standard deviation, describing the amount of certainty that a number has. And all of the numbers that you are seeing on the news and in social media have such a standard error or standard deviation describing the amount of uncertainty. And you are never shown that number. Never. Because it's large, especially for the case total, because of the inaccuracy and variance in these individual tests. So how are we going to understand anything? This is the question. How are we going to get to a understanding of this nonsense? Remember, this picture has been shown to us consistently on the news over and over and over again. This graphic was available on the internet in January in like 5 different sizes and has been used by almost every mainstream and indie media source since. It's not for nothing that these graphics were available. It's not for nothing that they keep showing you the same one. It's because they want to homogenize the experience so that neurobiologically it's actually homogenized among us. We feel the common experience. And staying home and being focused on social media has exacerbated that illusion of a common experience, an illusion. But it's not really an illusion. It's just that the coronavirus is not what we're collectively experiencing. It's the social engineering that has been hoisted upon us with the coronavirus used as an excuse. So how can we understand any of this? That's what today is about. How can we really understand how we got here and why moving forward the solutions aren't as simple as what are portrayed on television? And for anything, and I can't stress this enough, for anything, I believe, first of all, our individual community organization is, of course, very important. We need to get back in touch with our neighbors. I've been saying this from the beginning of my bike rides, starting in my first one. But more important, what I think is crucial is that in order to understand anything, you need to understand history. And I hope that there's a lot of people who are going, yeah, of course, I know, right? American history and yada, yada, yada. The history of money is very important. We're not doing that on this channel, but other people have. And it's important for everyone to know the history of money in America. It's important for everyone to know the history of your mortgage, the history of the money that you're using from your bank, and where it comes from, how it's generated. That fractional reserve banking is essential to understand. But I'm not doing that on this channel. What I want to do today is give you a brief history of viral biology so that you can understand how much we've known, the kinds of experiments we've done in recent history, not even deep history, but just recent history, the kinds of things that have happened in a little bit deeper history that have been blamed later, decades later, on laboratory experiments, and why the huge pile of circumstantial evidence is on the side of the scale of laboratory origin of this SARS-CoV-2. And why is that important? Why is that important? It is important to know where this virus came from to understand reality. As a neurobiologist, I'm going to explain one thing, and then I'm going to get on to the talk. Your brain is basically designed to do one thing. It's designed to take its past experiences, especially the recent ones, and try to best predict what's going to happen as you move forward in the future. On a small scale, that means your eyes have been trained to know what's going to happen when you move your hand and grab things. Your eyes have been trained, your hands have been coordinatedly trained through development to be able to predict what's going to happen. And when things happen that you don't predict, your body is surprised by it. If you grab this glass and it's hot, your body is surprised by it, drops it, and then puts it in its experience and says, wow, that's weird. Why was that glass hot? And then it connects whether it was on the stove or not. And the next time you pick up a glass, your brain will try to make a prediction about whether that glass is hot again. If you don't understand reality, your brain is trapped in a place that it doesn't enjoy. And you have a level of background stress that you should not have. That's called cognitive dissonance. It's when things don't agree, when like your boyfriend has lipstick all over his face and you're not wearing lipstick and he just came back from the bathroom and he says that it's from you. Cognitive dissonance says that can't be right. Those two don't exist in a world together. That's not possible. The world that we exist in right now, the largely electronically crafted information world that we exist in right now, including the one through which you are viewing me, is a world crafted almost entirely on agenda-based narratives that have nothing to do with the real history and the real facts of human civilization and the biology of it, and therefore we can't understand what's going on. And so today, first thing I want to do is bring you the history of this virus and the history of what's going on and give you some thoughts about why it's happening, and then preface what I want to do going forward in the future with the Saturday stream. So for me, this all started. I'm going far down in the corner now here. I'm way down here, guys. See me? Down here. This all started with a blog that I found in early February that was called Harvard to the Big House, and he had written a piece that I made a bike ride about because I was so fascinated by his ideas and fascinated by his insistence that this could indeed be a laboratory leak equivalent to like a Chernobyl type event, but then biological rather than chemical or nuclear. So this paper basically explains the history of it, gives you a few clues. But since I'm a biologist and I had access to PubMed and I read papers all the time, I started diving in deep based on the literature that he cited in this article. It's important to know that Dan's article is now a published bioessays article that I sent out as a PDF earlier this week. You will be able to find it later. I can send it to you later in the group PDF that I'll send after this stream is over. And in this paper, he discusses the things that I want to discuss today, but I'm going to discuss them in a bit more detail with a bit more of a bibliography. But essentially, if you want to read this paper and understand what I'm talking about, read it from a slightly different perspective. Dan's article, written by him and his father, is fantastic. His father used to work at Los Alamos Laboratory as an advanced biologist. The dude's also a genius. So he kind of served as a makeshift thesis advisor for Dan as Dan wrote this and did the research for this article. And I'm happy to say that this was finally published. It's worth your read, but that's what I'm going to talk about today is essentially, might SARS-CoV-2 have arisen via serial passage through an animal host or cell culture? And more importantly, why is that important to understand? And it's important to understand because we go back to the other half of the deception, right? If we understand the biology of it, then it will be very important to understand how the money that funded that biology came to exist and who decided that money would come to exist, because those are the people who are now benefiting from it coming into existence. So I'm going to explain the biology to you in a very rather objective way and let you come to the conclusion that I'm almost sure you will, that it's very likely that this is a laboratory-generated virus. And now the only thing is whether or not it's outside of the parameter space that, prescribed by nature, that our immune system is more than capable of handling on its own. At least from a population perspective. Maybe individuals, of course, like SARS and MERS, the originals, will have severe bouts with it. But this particular coronavirus is special. So, Dan's paper, please read it. This all started in January, and in the beginning of February, this paper came out by the now infamous Sheng Shili. She is the bat superhero, the virus superhero of China and the Wuhan lab. This paper came out on February 3rd, and it told us that the nearest neighbor for this virus was a bat covy RATG13 in their Ascension list. And they re-pulled this sample, and we carried out full-length sequencing on this RNA sample. Interestingly, just 3 weeks ago, this statement was corrected in a oral presentation by the Wuhan Institute for Virology, where one of their scientists corrected this and said that the sequence that they released in this paper was actually from a sequencing reaction that occurred in 2018. And at that time, this sample deteriorated and was no longer useful. So that's already a sort of reversal, a disingenuous moment here, because this is a Nature paper. It's a Nature manuscript coming out purporting to explain and identify the pathogen, which was at this time just starting to be acknowledged as a likely pandemic. So this is very crucial, because only after our pushing of this data out was this seminar instigated, and then in this seminar did they address this issue, only because we brought the contents of this talk out. If this would not have been pushed by Dan and myself and many other people into the more mainstream discussion, a lot of this data might never have been mentioned and never discussed, and the zoonotic story might never have been seriously challenged. I'm not taking any credit for what I've done. I'm taking credit for being part of a wave of people that have made this happen. So this paper is cool, and it's important to understand that we've been fooling around with coronaviruses as another entity for a very long time, because coronaviruses are incredibly safe. We have known about 4 coronaviruses that are endemic to the human population, 4 of them since about 2006. We actually only sequenced the 4 respiratory viruses, including the 2 that are blamed on or the one that's blamed on the common cold, only a few years ago. These are not entities that we have understood very well for decades. These are entities that we've only been able to identify less than a decade ago. That's important to understand. Because when we identified them as an entity which did not evoke the systemic immune response that measles or polio or many of these other severe diseases evoked. And in fact, because it doesn't evoke symptoms in many of the victims, the infected individuals, this paper describes why they would be a very great candidate for a multi-gene vaccine, a vaccine for other things like HIV. And the reason why is because since they don't evoke an immune reaction on their own, most people get a common cold and they don't even know they have one, or they just work through it. Then if you could have a common cold carry a protein which imparted immunity to another pathogen with the same common cold, this would be the ideal vaccine. This idea is from the year 2000. Now, the long history of laboratory leaks has been documented quite well lately. This article is from May 2020, but we can go down a long list of articles. This is from 2015. This one is from 2014, written by a guy from Harvard talking about influenzas and the enrichment of influenzas and why the heck are we doing that when we already have dangerous influenzas. This is Lynn Klotz and Edward Sylvester. These two authors have written a number of articles, including this one, about the consequences of laboratory pathogen escape, either infection of laboratory workers or unwitting escape via animals, waste, whatever. In fact, the number of articles written and covered, the amount of data that's available is overwhelming. The history of biological research around the world, mankind has never been flawless. Mankind has never been able to do this kind of research without accidents. Never. Just like nuclear power, you can't do it without accidents. You can't fly airplanes without them crashing. You can't drive automobiles without them crashing. You cannot manipulate pathogens without there being accidents. This is Mark Lipstickskin. More importantly, to understand very specifically, there are well thought out scientific arguments and analyses available that describe various previous pandemics and epidemics as likely laboratory originated pandemics or outbreaks. The 2009 swine flu has been debated for a long time as being a laboratory produced virus. We know that we've made avian flu many times in the laboratory. We know that avian flu can generate itself on animal farms. So the animal farm viruses have never really jumped. It's never really been shown. But we've generated a lot of them we've generated recombinant flu in ferrets in 2011 and 2012 in laboratories in the Netherlands and in the University of Wisconsin-Madison. These studies created a global outcry. What in the world are you people doing? Why would you make a pandemic flu virus that didn't exist? What would be the point? So this paper came out in the beginning of March. It's called The Proximal Origin of SARS-CoV-2. This paper is cited repeatedly, still to this day, as the seminal account of why the virus is not, cannot, would never have come from a laboratory. The primary argument that's made here is that no one could have imagined or engineered this sequence. We're not creative enough. We don't understand proteins and RNA and DNA and protein function and structures of proteins. We don't understand it well enough to select this sequence to make such a pretty virus. And Dan's point, my point, and the point of everyone who is arguing against this nonsensical paper that really proves nothing and argues really nothing. In fact, it cites papers incorrectly. It says that papers show things that they do not show. the virologist from Norway, his paper is excellent at describing this, which we will probably talk about next week. So the theories of the SARS-CoV-2 origin that are listed here, it is improbable that SARS-CoV-2 emerged through laboratory manipulation of a related SARS-CoV-like coronavirus. They are starting their summary paragraph about the theories of SARS-CoV-2 with a sentence which specifically denies the most likely source. This is a paper which had not acknowledged, was trying to hide, and even pretend that the minors and the outbreak that occurred in 2012 did not exist. It was not wide knowledge at this time when this paper came out. It is even possible that Christian Andersen did not know about that outbreak in 2012. Did not know about those papers. It's possible. Because like all other biologists, I'm afraid that Christian Andersen is also fragmented in his head. He knows a lot of papers that are related exactly to the things that he did in his PhD and exactly to the things that he did in his postdoc. But all other related research lines, all other related biological questions are outside of his expertise, and he's fine not to speak on them. The general biologist in the world is dead. They're gone. They don't exist anymore. Everybody hides behind their own little corner of expertise. And if you work on sarbocorbal, whatever viruses, you're not willing to speak on flu or on coronavirus because, you don't know enough. It's not my specialty. This is also the dilution of intellectualism in America. It's the destruction of intellectualism in America. I'm a general biologist. I know enough about how cells work and how mammals work and how the brain works and circulation works and the immune system works that I can add to that and continue to learn about it. I don't need to be a specialist. You need to be a generalist. And this specialist is trying to tell you with that first sentence, it is improbable that SARS-CoV-2 emerged through laboratory manipulation of a related SARS-CoV-like coronavirus. That sentence is exactly the opposite of the truth. The truth is that it is probable that SARS-CoV-2 emerged through laboratory manipulation of a related SARS-CoV-2 coronavirus. That's the exact truth. So the reason why I think that's true is because all of the aspects of this novel coronavirus that have been hyped on the news, including the cardiovascular effects, including the neurological effects, including the cytokine storm effects, including the anything and everything, the multisystemic inflammation syndrome, the Kawasaki syndromes, which is actually multisystemic inflammatory syndrome in kids. All of these things that are portrayed, thrown at you in that fire hose of information, they are thrown at you as new, novel aspects of this coronavirus that we've never seen before, as a way for you to try and start to believe that we know very little about the biology of this coronavirus, and therefore we should take all precautions necessary to survive. It may kill all of us. That's the logical path they're trying to take you down. That's what they want you to think about. You just can't know. We can't know. So we've got to just take the most precautions necessary. It's a very lawyeristic view of the world, isn't it? It's a very lawyeristic view of the world, where actually we have to be afraid of everything just in case it happens now. We can't open schools because kids might get sick. Yes, that's biologically true. Kids will get sick. A lot of them might get coronavirus and test positive for the RNA. But the numbers around the world tell us that kids aren't in danger. That's the truth. But before I go there, sorry that I got off track. I almost lost it. The novel aspects of this coronavirus are not novel. You need to understand that right now. We knew already we knew that the cytokine storm plays a direct role in morbidity and mortality for influenza and for coronaviruses. We knew that a long time ago, especially for the severe SARS patients. We knew it was a cytokine storm. So the cytokine storm is not a surprise for this SARS-CoV. And the cytokine storm is also very rare among healthy people, if not completely unobserved. Do you hear that? It's rare if completely unobserved in healthy people. We've also known for many, many years that these bat viruses, this is Sheng Li Shi's paper, we've known that these bat viruses are out there. They've been searching for them for years, collecting them for years. But we have known that the antibody levels to coronaviruses go down after a year. We've known it for the MERS. We knew it for SARS. It doesn't seem to matter. That's important to understand. It doesn't seem to matter. And the reason why it doesn't matter is because there's T cells behind this, right? If the antibody titers go down, it says nothing about your T cells. It says nothing about your memory B cells. It just says that your circulating titers of antibody go down after a year. This is not new information, folks. This is old school. We knew this a long time ago. This is an antibody that is causing antibody-dependent enhancement or some type of cytotoxicity as a result of developing antibodies with to the spike protein. So what you're looking at here is the occasional patient that at SARS-CoV-2 developed anti-S2 antibodies, and they were causing cytotoxic injury, enhanced immune cell adhesion to epithelial cells, and autoimmune responses. So this is also not surprising that this variant of the SARS family would occasionally cause antibody-dependent enhancement. Even more important, though, we know from papers in China that the origin of the original SARS virus was also suspected to be from a laboratory, because they were also unable to really find a convincing zoonotic trail. And soon after the original SARS outbreak, several more times SARS was re-released from laboratories in Beijing and Taiwan and elsewhere. So the whole debate has really its roots in the concept that we didn't know, that we don't know. This is all brand new, and it's not. And it's also rooted in the idea that we could have never made this virus. That's what Michael Osterholm said on Joe Rogan in February, and what made me go nuts after this Christian Anderson paper came out. Almost simultaneously, I think that happened. That he went on Joe Rogan, and then the Christian Anderson paper came out, where they say it's not likely or improbable or impossible that it came from a laboratory, while spelling out exactly how it came from a laboratory via animal passage. And the reason why animal passage is real is because we did that already with the flu. I showed you those papers already, and I'll show them to you again. So what's important to understand here, I should have put a break slide in here. I'm apologizing for that now. See if I can go here and give you a break from that. So what's important to understand here is that to say that we could not have designed this virus is not to say the same as that we could not have made it. And I think it's important for you to understand how long the technology exists that we could make viruses from scratch. Meaning that if we had a computer sequence, a text file that described the protein sequence of the virus, already in the year 2000, we had the technology, the gene transcription, the manufacturing, nucleotide manufacturing technologies to create a poliovirus from scratch. A poliovirus is 7 kilobases long, 7,000 amino acids long. And in the year 2000, we proved that we could create the polio virus from scratch, from a sequence. And remember that the idea here is very complicated because DNA in your cells gets translated to RNA in the nucleus. It gets tagged funny, and then it gets extruded out of the nucleus, and out here in the cytoplasm is where the cell translates the mRNA into proteins. The mRNA in your cells is tightly regulated. Foreign mRNA is identified and destroyed. mRNA that's been around too long gets destroyed. And the body and cells have very complex ways of regulating how long particular transcripts of mRNA can exist inside of a cell and continue to be read before they get thrown in the garbage. That's how your body regulates the amount of proteins that are expressed in cells. Viral RNA is inserted into the cell and uses your own protein manufacturing machinery to create more copies of itself. And so the virus has to have very complex strategies to avoid detection inside of your cells and use your own machinery to its advantage to make more copies of itself. That's the principle of viral replication. The trick here is that mRNA, as I said, RNA in general, is very unstable. And most of the enzymes are not designed to copy RNA. Not only is it unstable, but the enzymes that copy RNA are also notoriously bad for making errors. Whereas the enzymes that make DNA and the enzymes that proofread DNA are very, very, very high fidelity. So the trick is, and what happened in the year 2000, is that we came to the point where we could make an mRNA, an RNA-based virus using our DNA tools. Because our DNA tools have been very easy to develop. And over the last 20 years, we have developed DNA tools that basically make it possible for us to create any sequence from scratch. The question was, in the year 2000, if we did that with a sequence that represented viral RNA, and we allowed that viral RNA to be transcribed inside of a cell culture, would that viral RNA result in the production of viral particles? In the year 2000, we did that with polio. And in the year 2002, Ralph Barrick's lab did that with a full-length infectious cDNA of mouse hepatitis virus strain A59, which is a group 2 coronavirus, around 31,000 kilobases in length. So in the year 2002, published, so he probably did it in the year 2000 or the year 2001, Ralph Barrick's lab published the creation of a coronavirus from scratch. And in this paper are a number of crucial methodologies that show and demonstrate or describe how to take these shorter pieces of this extremely long sequence, chain them together. Later, Barrick's lab developed ways to seamlessly ligate these pieces of DNA together so that you could get exact sequences, and then to transcribe this into infectious RNA and then grow it in cultures. This was in 2002, 18 years before the coronavirus was released in Wuhan. A year after this paper was released in a more obscure journal, the same lab published a paper where they showed that they could resurrect a SARS-CoV-2 from 2003 that was no longer available in nature and only available in translation text form. This is in 2003, folks. They can pull out a text library, and with a series of methodologies, and well-developed methodologies, they can bring us a coronavirus from scratch. I hope you see where this is going. There are numerous reviews. This is a 2014 review reviewing the coronavirus reverse genetic systems, which allow you to manipulate coronaviruses, exchange pieces of coronaviruses between viruses, and to transcribe viral sequences and alter viral sequences from scratch. CRISPR only contributes to this, right? CRISPR-Cas9 can be used to edit DNA. Early in this debate, people were arguing that, CRISPR-Cas9 can only be used on DNA. But in 2002, we were using DNA to make infectious RNA virus. So who cares? We'll do it. We'll use CRISPR-Cas9 on cDNAs that encode for viral RNA, and we'll get infectious RNA. We showed that 20 years ago. So again, that's another disingenuous argument made in media about why this laboratory hypothesis is nonsense when that argument is nonsense. Oops, we're going the wrong way there for a second. So this is a synthetic recombinant bat SARS virus in 2008. This is just basically a better example, an advanced example several years after that 2003 paper, again from Ralph Merrick's lab, showing you that they can create a completely synthetic virus, one that never existed in nature before, and it'll work. It can infect cells. They can play with it. They can grow it. So this is the paper that everybody got really excited about at the moment that this came out and said, oh, look, they did it. They made it. And then everybody said, what are you talking about? This has nothing to do with this. But it does. This paper has something to do with it because what this paper shows and what this bibliography of this paper shows is what they're capable of doing. What's important to notice here is that this paper selects a SARS-like virus, SHC014-CoV, and they fool around with it by exchanging it changing its spike protein. So swapping the spike protein of the SARS virus onto this one and using chimeric manipulation to put spike proteins from one onto the backbone of another. The reason why this is important is not because this virus in particular did this. The reason why this is important is to realize that in this paper, the virus that they have selected was selected from a pile of viruses, a huge sampling event that happened in 2012 and 2013. Most of the papers that are published by the Wuhan Institute for Virology in the last 8 years running up to this pandemic have been based on samples that were taken between 2012 and 2013. That is 100% true. They pull them out of the freezer and they sequence them. That's what they say. More likely, they're growing them in animals and in cell culture. And they've been doing it for years. This paper shows you what they can do with an innocuous example of this kind of experiment, where both sides of the equation are relatively innocuous viruses and we can swap pieces. The only other difference would be then that let's take a more or less innocuous virus and swap pieces with another or less innocuous virus and see what happens. And these experiments may not have been published, but this paper tends to suggest that they are done, as does the admission made earlier this month that they have lots of sequences that they've never published because they're intellectual property. So this is the same paper. Sorry, I don't know why that's up there twice. In this paper, one of my YouTube viewers noticed that there were several discrepancies between the original online bibliography and the one that was published formally on Nature's website. Those papers are interestingly omitted in the sense that they provide some intellectual background for understanding how much they knew. That's right. Had what they knew. I see. I'll try that. Okay. Okay, so now I'm just going to break quickly from this to say that I'm going to activate screen sharing on the Zoom to see if that changes anything for you guys. Start sharing. And I'm going to share desktop 2. Share. And so now the Zoom meeting is also sharing my desktop. And we're going to briefly see what that does to the screen and to the stream, because that's going to change things. If it changes the stream, I'm going to stop sharing. So give me feedback immediately on that if you can. So this paper left out a very important detail, 5 very important details. If you think about what this paper meant and how much scrutiny, this paper was given, these 5 papers are important to understand because they give you a very clear idea of how much they knew about the investigating they were doing. This reference from that paper that is omitted shows us that they know that there is a very, very unique way that the receptor binding domain of SARS-CoV-2 determines how infectious the virus is. And they look at how efficiently the efficiency of infection is based solely on the spike protein. This is a discovery in 2007. So in addition to that, they're also looking at the amino acids in the receptor binding domain that are important. And here they identify amino acids 310 through 518 as being responsible for the binding to ACE2. That is also a 2008 paper that is omitted from that. There is a 2005 paper that is omitted from there, where we look at 2 critical amino acids that are responsible for changing the binding to ACE2. So far, we're seeing a lot of evidence that they know a lot about how the spike protein binds to ACE2 and how important that is for the infectivity of coronaviruses. And this is 10 years before the emergence of this pandemic, and it is 10 years after the papers where they show you that they can make coronaviruses from cDNA from scratch. We also see papers omitted where they show that they are studying very specific proteins. This is the open reading frame 6 protein and studying what role it plays in infectivity and viral copy number. This is an important paper that they also leave out of that. And they leave out this chapter which actually describes the methodology that's used in this paper. This paper specifically used human airway cultures to enhance or enrich the viruses that they were studying. And you can imagine a scenario where human airway cultures would be ideal for enriching for a particularly virulent respiratory virus without needing animals, and do it very rapidly, very cheaply. The other thing that looks at here is that they actually showed synthetically with this paper that they can watch the change. They can actually predict if we change this, we can get this civet virus that doesn't infect human cells to infect humans, ACE2. I think these are mice, but the point is that they have a mouse model of the ACE2 receptor, and with a small change to the civet spike protein, they show that they can get this to happen. They're also learning about how these changes occur and how they can make it happen through a dual tropism sort of change. It's a complicated paper that essentially reveals that in 2008 they already knew the molecular mechanisms that lead to zoonosis, and they knew how to harness them using animal passage. So this is just an example again of the fact that they've known that this is going out. This is a paper again that's drawing from those viruses from the same caves that we've talked about for a long time. Another one, paper about caves in the Yunnan province. This is a different cave than where the outbreak happened, but because their record keeping is so terrible, it's very difficult to know for sure which viruses came from where, but in general, you can assume that most, if not all, of the viruses that are currently being worked on as sequences are being drawn from these viruses that were found in the Moyang County of Yunnan province in 2012. That's also a paper about these bat viruses that are being found in caves. All of these papers have come out before the pandemic. And at the same time, lots of people are trying to make the argument, and have been making the argument but being ignored, that this is a laboratory manipulated virus. One of the best ones is This is also a paper that came out, to show you that the spike protein is odd because it contains this furin cleavage site. And that's one of the things that Dan's paper really analyzes very well. And I don't talk about very much today, but we'll talk about next week a little bit more. But it's just one piece in the puzzle. And this is often eliminated during zoonosis. It's not really clear why this would show up here, but it shows up regularly also in other enriching experiments for flu. And so we've seen the sudden appearance in laboratory experiments of a furin cleavage site or a multi-basic cleaving site. And so it is often indirect evidence, or at least seen as indirect evidence of laboratory enrichment. But in this case, they're finding or trying to purport to find zoonotic evidence of the generation of this, but none of those accounts are really working. This Nerd Has Power blog came out and really hit a home run in terms of identifying one of the problems with the RATG13 virus, which we talked about earlier as being the closest related sequence in their database that they reported in February. The reason why this is interesting is because after careful reading of the literature by people on Twitter, I was also reading, but I didn't find this detail. This paper describes the mine shaft in Moyang County. You can see it right here. In Moyang County in Yunnan Province, China, from 2012 to 2013. It is from these surveys, these trips to the caves, from which all of our data about viruses related to SARS-CoV-2 is derived. And interestingly about this is that this paper purports that there were 3 miners which had a pneumonia. I don't even know if it's this paper. I think it's actually another paper. I'm sorry that let's see, 6 bad species. No, this paper doesn't even talk about it. There's another paper related to this survey which shows us that this is the paper that shows us that there was a outbreak where 3 patients, read carefully, 3 patients who were working in a cave got severe pneumonia and died. And then this paper and this paper are the result of surveying those caves for the next year and a half during the off season of bats when they were hibernating and whatever. So keep that story straight in your mind, right? Because we are talking about viruses that were sampled from a very specific cave in which, according to this more obscure publication, 3 patients got severe pneumonia and 3 patients died. This is another paper that comes from the same place, describing some interesting idiosyncratic viruses that were found in the same cave. The Moyang virus attachment glycoprotein paper. At the same time, we saw a description in June in Scientific American where they interviewed Shengxi Li, and she talked about the cave viruses. She talked about being called in because of a severe pneumonia outbreak. But in this article, they talk about the wrong cave. Specifically, they never mention the Moyang province, ever. They never mention them. And that is a clue that they are trying to throw you off with this. This one also is a very good article to read, but again, this article is throwing you off. It's not talking about the cave that is in the Moyang province, and it's not talking about the cave where there was a pneumonia outbreak. It's talking about a different cave where a lot of those other viruses came from. And this is a falsehood. They want you to ignore the cave in the Moyang province. But fortunately for us, somebody found the original documents. We were able to download an entire master's thesis which described the outbreak, described the 6 patients who developed the pneumonia and 3 patients who survived. The master's thesis was translated and published. The translation of this master's thesis indicates that indeed there were 6 patients, 3 died, 3 recovered, and bronchial lavages were taken from all of these people, including the cadavers. Those bronchial lavages were sent back to the Wuhan Institute of Virology, where no publications were ever made from them again. No data from those samples has ever been released. No data from those samples has ever been published. No data from those samples has ever been acknowledged since the outbreak, since the pandemic started in January and February. Even in the live stream discussion that occurred 2 weeks ago, chaired by these virologists in Wuhan, none of them acknowledged this cave. None of them. And when they did acknowledge this cave, they did not acknowledge the sampling of the miners. But this master's thesis shows you that they know they were treated, they were diagnosed with the same symptoms as COVID-19. They were treated the same way as people with COVID-19, as having an unknown viral pneumonia with ground glass opacities in their lungs. And at the same time, we are now left with biologists around the world who are not fearful, are showing us that there's something very special about this virus. Nikolai Petrovsky's paper from Australia shows us that the binding affinity for the human ACE2 receptor is so exquisitely high that the natural origin is dubious. Natural viruses don't need to develop affinity of this magnitude. They generally don't do it. And the interesting part is that despite having such high affinity for us, the overlapping affinity with other animals is also quite high, which does not suggest that it's a zoonotic virus. It suggests that it's somewhere outside of the realm of nature, because natural viruses don't often have pan-infectivity. In fact, they rarely, if ever, do. This might be the first virus with pan-infectivity in mammalian species. Another odd signature. Helena Chan's paper, she's at the Broad Institute at MIT. She did an extensive analysis of the sort of the evolutionary change as evidenced from the sequence change. We've been sequencing the virus, purportedly sequencing the viruses that are collected around the world as the pandemic has moved through the planet and our population. And so the virus is changing. And what Helena Chan's paper shows is that the virus is not changing at nearly the same rate as the original SARS virus did when it was released and its sequence was charted over time. The original SARS virus had about a 6-month period where each individual strain was developing very rapidly. Many of them disappeared. The infectious ones were followed until the end when it was gone. And it was late phase viruses were changing less than early phase viruses, and there is no early phase in this virus. It's all changing very slowly, which is serendipitously very, at least on paper, valuable for anybody who wants to make a therapeutic or a vaccine for this virus, because it's not changing that rapidly, so you've got time to develop your profit-making intellectual property. And this is a picture from Helena Chan's lab to try and, from their paper, to try and show you at least the hypotheses on one side at least fits some narrative better. The left side is the original SARS. The SARS-CoV-2 is just automatically already adapted for us. And so even a hypothetical, which doesn't really seem very good here, even the one on the left is not that good. The one on the right is just missing everything. We just don't know where it came from, period. You can put a bat up there somewhere, but that's very disingenuous. There's just, there's too much unknown in between whatever came before it and us. And that's the point of these papers. There's just too much unknown. There's not enough data to support the zoonosis, even if that's what a lot of these virologists insist is happening. There's just not a lot of data to show, because a lot of the evidence is missing. So, interestingly enough, there are all kinds of anecdotal things that you can find in the history of the Wuhan Institute for Virology that will also not help the argument that this is not a laboratory virus. It won't help support the argument that they're so good at biosafety level 3 and 4 that this could have never been released. Here is a patent application from the Wuhan Institute of Virology for a very fast applicable tourniquet for when you get bit by a laboratory animal. Here is a carnivorism bat rearing cage. So one of the things they tell you in the mainstream media and on social media regularly since the beginning of this is that they only sampled fecal samples and took those fecal samples back to the laboratory and studied those viruses. They never had any live animals. They wouldn't take live animals back to the laboratory, and they wouldn't use live animals in experiments. But we used live animals like ferrets in experiments already 15 years ago to enrich avian flu. There's no logical argument to say that we wouldn't be using live animals to enrich or to at least study coronaviruses. That's not a genuine argument. That's completely false. And this is another example. This is a application for a patent for a specific bat rearing cage, which shows that they have common circulation between them. And if they have common circulation, then you're seeing the possible enrichment for airborne viruses in bats, just like they did with ferrets and avian flu in 2012. How is the stream going? Can anyone tell me, is the stream going okay now? I'm going to stop sharing the thing on Zoom for a minute, and then hopefully somebody will tell me what's going on here. I hope that the adding the Zoom didn't slow down the stream, but that's one of the reasons why I stopped it on the Zoom. If someone can tell me whether that's the case or not, that would be great. Did the stream slow down or not? I don't see a chat window here because this is just annoying, so I'm wasting. I don't want to do that. Sorry. We're going to try and troubleshoot the Zoom when we do questions. No effect at all? Oh, great. That's awesome. Then I'm going to start it again. Share screen again. That's fantastic. Okay. I'm going to start this sharing of the screen again on the Zoom for you people. So what we're dealing with here is there's just too much circumstantial evidence. Billy Bo Stickson on Twitter has been working countless hours. I don't know what drugs he's on. I don't know if he's actually an AI or what he is. But he has been compiling evidence in different threads of all different things. One of the most plausible explanations that I see here is the mismanagement of waste disposal at the Wuhan Institute of Virology, especially if they had animals in the same facilities. The reason why this is important to understand is because if they had animals there and they were culturing viruses in various animal hosts, then they would have animal waste, they would have dead animals, they would have sacrificed animals, they would have animal materials that all needed to be properly disposed of. If they were not properly disposed of, the possibility of laboratory escape increases many, many fold. And in fact, the problem of waste disposal was well documented during 2019 at the WIV, and there's lots of evidence for the fact that they were aware of the fact that they had some problems and that the system was not foolproof, despite it being a biosafety level 4 laboratory at the maximum state. So this is disconcerting, folks, because there isn't a lot of reason to believe zoonosis given this intense level of information. This is another paper that I think you can read and try to digest, the origin of COVID-19 and why it matters. And it matters because we need to understand this. This is, again, Nicholas Petrovsky's paper. Am I going backwards here? No, sorry. I'm reminding you of that again. This paper is from India. It is a controversial one. It is not one that's going to be met with a lot of open arms. But what this paper basically shows is it makes a direct correlation between body mass index and the severity of SARS-CoV-2, or the severity of COVID-19 infection. And they make a correlation between body mass index, and their explanation, their hypothetical explanation for this is that obesity is a chronic inflammation state which biases your immune system toward the cytokine storm and over-inflammation, exaggerated inflammation, and prolonged inflammation. And so then obese people are more susceptible to these compounding effects that have been hyped, the anecdotal presence of which have been hyped. And this paper leads me to believe, and a couple other papers lead me to believe, that if they told you the truth about the people who are having the very severe cases, they are not healthy individuals. They might not smoke, they might eat pretty well, they might not have any chronic illnesses, but they could be 25 pounds overweight, and that might be enough for them to be in a slightly inflamed state all the time and put them into a higher risk group than what you think of in your imagination as a healthy American. And that's what this is about, the cytokine storm, and the fact that interleukin-6 in particular is playing a central role, and that adipose tissue has interleukin-6 receptors, and so that they can effectively dilute the interleukin receptor. I lost my camera. I'm going to have to switch that. Hold on a second. They could have lost the drive for interleukin-6 because you can imagine a scenario where interleukin-6 is, if you imagine that scenario, interleukin-6 then is in a is the primary sender signal for the T cells. And in that scenario, when the T cells are sending this interleukin signal out, it goes and it finds receptors and then binds to those receptors. If you have more percentage of fat in your body, you will have more percentage of interleukin receptors that don't do very much. And when they don't do very much, that means that the T cells that are responding to a particular infection need to release more interleukin-6 in order to reach the threshold of signaling required to evoke the immune system response that they themselves are coordinating. After they stop calling in the reinforcements by releasing interleukin-6, the interleukin-6 concentration starts to go down in your plasma, but it is sustained longer in a fat person, in an obese person, than it is in a healthy person, because there is interleukin-6 being released from the receptors in the adipose tissue that sustains the concentration of interleukin-6 for a longer period of time after the T cell response is waning. This is a feedback loop called the cytokine storm that is exacerbated in obese people and is probably, very likely, the most parsimonious explanation for why we are having more deaths and more severe anecdotal cases than other countries around the world. So the question now becomes, as we move forward in this thing here, what time is it? It is 12:21. I guess I've been talking for a while. See, this is good. This is not going to be a problem. I like that a lot. We're going to have a question and answer session in about 10 minutes. So how much biology is left? I think there's a lot of biology left to fix. I think there's a lot left for us to talk about, and that's why this Saturday stream is going to become more and more polished. More and more am I going to dedicate my weekly time to making sure that this stream is a fountain of useful knowledge, not information that may or may not be true, but useful knowledge. That's what information is. It is information transformed into a useful form. And what I want to do in the coming weeks and months is start to provide you guys with reliable information that will allow you to look straight into the fire hose of information and see it for truth. Because right now, if you're trying to look on Facebook and look for your Facebook friends and look for the Facebook trends and look for the Instagram, Twitter trends and try to understand what's going on in the world, you are fighting against something you cannot beat. And that's what that Shadowgate viral video is about. That's what all of this conspiracy stuff was about for the last 30 years. Media is being used against you, and there's more media present in our lives now than ever. And so there is more false information present in our lives than ever. And so the things that we need to think about moving forward are manyfold. In the short term, what I hope to give you is a firmer understanding of viral biology in general. This is not going to be enough. This is just the start. This was just the history of what we've been doing for the last 2 decades, and why saying that we could not have made this in a laboratory is absurd. That's all I gave you today. I didn't prove it to you. I just showed you that common sense tells you that it's not absurd, and will help you to question the motives of the people who jump to that side so unequivocally, without question and without critical thinking, without backing up their jump. Because those are the people who are using this viral biology against us, not to open schools and to keep the lockdown going and to tell us that we all have to wear masks and to scare kids into thinking that if they go back to school, they're going to bring the coronavirus home and kill their parents. If that's true, then we're never opening schools again, don't you see? This cognitive dissonance is a prison closing around you that only sound biological information, sound biological knowledge, and sound financial knowledge will allow you to escape from, or at least minimize the damage that it does to you and your family. That's what peak prosperity is about. His website and his goal is to help people survive the coming storm. And the coming storm is being created by falsehood and by confusion and by stress and cognitive dissonance. We are monkeys, folks. We behave like monkeys. We don't like to be put under pressure. We don't like to be afraid, and we don't make decisions very well when we are afraid and confused. And so my goal is to help you become less afraid and to help you become less confused about the biological aspects of our world. I want to talk about virology in general. I want to give you a real life model of the immune system next week. And I want to give you a real life model of what vaccines do, what the concept of vaccination is, what an adjuvant is, why they need to use it, why the selection of adjuvant is important, and why the wrong adjuvant has been shown over the course of many decades to be a problem and likely the result or the cause of vaccine injury. I want to talk about nutrition. I want to talk about malnutrition in America, and I want to talk about the consequences of malnutrition. I have a very, very intelligent and insightful viewer who has cued me into the fact that there's actually a whole literature of veterinary nutrition which sits in opposition but parallel to human nutrition in mainstream media. And in fact, what he opened my eyes to is the idea that if you read veterinary literature about how to take care of livestock for maximum yield, for minimum health costs, you will find advice which is directly in opposition to the mainstream ideas about human nutrition and about human fast food and about human production of food, about school lunches and about school nutrition. All of these ideas are contradicted by veterinary information about livestock. And this contradiction is a false one. What's good for animals is good for us because we are animals too. What makes a cattle's immune system strong so that your entire herd doesn't get sick is the same thing that will make us strong. And that's what I'm going to bring to you in the coming weeks. Exercise and the consequences of exercise. This cannot be underestimated either. I'm going to bring you data, primary literature data about how exercise contributes to health and how exercise contributes to mental well-being. Why it's important that when we don't exercise that all of these other things snowball behind it. You can be well nourished, but missing exercise can still make it possible for you to be metabolically unhealthy. And finally, my specialty, which will always be in the background and always be the tint or the angle that I will present biology on whenever I have the chance, because that's what we are. We are brains. We are brains in a body, right? If you love your wife for the good reasons, you love her brain. You don't love her boobs. You love her brain. And when we love our best friend, we love their brain. And so understanding how our brain works in order to extend well-being to all is the ultimate and primary goal of this channel, of this stream, of Gigaohm Consulting going forward. And so this stream will be every Saturday at 11 o'clock, and it'll go until about 12:30 or 1. At around 12:30, what I'm going to try and do is open it up for questions. Right now, we're going to play around with that a little bit. If you have any questions, now would be a good time to log on to the Zoom meeting or to bring the Zoom meeting to the front of your desktop. Just one last slide before I open up this to questions. One of the interesting things going forward now is going to be thinking about aspects of our life and aspects of our socioeconomic sort of civilization, community that we live in. What aspects of our culture could possibly be contributing to the manifestation of coronavirus in America? One of the things that could be contributing to it that has been forwarded by many people is the use of the flu vaccine in America. In case you're unaware, the use of the flu vaccine in other countries, especially that I am aware of, the countries in Europe, is a very focused use of the flu vaccine in places where people are vulnerable. They use it on old people. They use it on people that work in hospitals, and they use it for vulnerable people. They do not, in any way, shape, or form, in the Netherlands, for example, distribute the flu vaccine in local pharmacies like we do. They do not distribute it at schools. They never give it to kids at all, which is interesting because kids are quite susceptible to the flu. And for a small percentage of kids, the flu is much more dangerous than for the small percentage of kids for whom COVID-19 will present a severe case. None of them are dying. They do die of the flu. This is a paper, and there's another paper from looking at Army soldiers inoculated with the flu virus. And both of those papers show that there is a higher rate of other respiratory viruses, including rhinoviruses and coronaviruses, in people who have received the flu vaccine, and only a mild or no significant decrease in flu infection. In this paper, they are showing that the risk for influenza does not significantly decrease in kids that get the flu vaccine, but the kids who got the flu vaccine have a very significant increase in the manifestation of coronavirus and rhinovirus respiratory infections. So one of the possibilities here is that the flu vaccine has created a propensity for some people to be sick from from SARS-CoV-2. This possibility is very difficult to investigate because there are a number of producers of the flu vaccine, there are a number of variants of the flu vaccine, and there are infinite distributors of the flu vaccine. And so it would be very difficult, if impossible, to track down who's had the flu vaccine and who hasn't. Because your doctor is often not the person who gives it to you. It's not in your medical record. It's not there for everybody to see and make a correlation to. And that's an unfortunate thing for us, but a quite convenient coincidence for those people making the billions of dollars in profit on the therapeutics and potential vaccines that are currently being manufactured. So this was Gigaohm News. This was High Resistance, Low Noise News brought to you by a biologist. My name is JC. Oftentimes I am JC on a bike. Later this afternoon I'm going to release a bike video which covers this same bibliography and in a little bit different light, and very specifically it does it on a bicycle, so it might be a different sort of presentation mode. It's a different sort of discussion. I have less notes in front of me there, so the the stream of consciousness on the bike tends to be a little bit more solid. And also, of course, watching someone bike while they lecture about the origins of SARS-CoV-2 is also quite entertaining on a different level than the live stream is. So hopefully this afternoon, some of you will be able to check that out.